All right, so here we are going to talk about mitosis. Okay, there's a joke about this too. You know, what did the chromosome say when his sister stepped on his feet? Mitosis. Anyway, they're a little funny for you. All right, so we're going to do this as quickly as we can. The first I can statement is I can describe mitosis, and this is talking about how it looks and what it is. And I'm not going to go into how a cell looks in mitosis because I'm going to look at each phase separately. So you'll see that as we go through it. But basically describing mitosis is it is division of the nucleus. Okay, when you're producing two genetically identical daughter cells, it is the division of the nucleus. And so the first phase, and I kind of almost shudder to say the first phase because these phases really do blend right into each other. It's not like prophase ends and then metaphase starts. They kind of, as prophase is wrapping up, metaphase is already starting. All right, but in prophase, what do we tend to see is we tend to see a couple of things happening. Whoops. We tend to see our centrioles move into opposite ends of the cell. We see our nucleus dissolving, so you're starting to see that nuclear envelope break up, and we're starting to see our chromosomes condense. Our nucleus almost starts to look noodly, and that's one of the ways we know we're going into prophase, is those chromosomes are basically shrinking up and condensing. They're supercoiling and all that so that they can move. It's a whole lot easier to move like a ball of yarn than if you unwrap that whole ball of yarn and have to move that pile of yarn. So that's basically the concept, is to get it condensed so it's easier to move. All right, so prophase, nuclear membrane dissolves, chromosomes condense, centrioles are moving, and spindle fibers are starting potentially to form. Okay, then there's metaphase, and I hesitated for a minute because AP likes to include pro-metaphase. Pro-metaphase is just when it's further along. Wow, I don't know why I made that so big. But anyway, it's a little further along. So the nucleus is more dissolved, the chromosomes are more condensed. These are probably already at the opposite ends and making spindle fibers, yada yada. So pro-metaphase is just what you would think. It's in between pro and metaphase. Cell does not get bigger. I just did a really bad job of trying to draw the same size cell. But what we see in metaphase is we see that our chromosomes, and I'm only going to draw 4, but know that in you this would be 46 chromosomes lined up in a single file line. They get to the middle, or they're kind of in the center-ish of the cell just because that's where they were when the nucleus dissolved. But what happens is there are some microtubules that grow out from the centrioles. We call these spindle fibers. And they're going to attach to the chromosome at the kinetochore. So when you look at a chromosome, a duplicated chromosome, there it is. There's the centromere, which is the point of attachment. And then there are these little brackets on either side of that, and that's the kinetochore. That's where the microtubule latches in or locks in. So this microtubule is going to grow and lock in on this side, and this one's going to grow and lock in on the side of that one. Okay, and so all of those are going to lock in. Then you're going to have ones coming out of this centriole. They're going to lock into the other side. And that way when they divide, half will be pulled each direction so that you get your nice, happy daughter cells. So one of the things that's happening here is we're making sure that it's locked in on both sides, and once it is, these two kind of play a tug of war to get these lined up in the middle. So that's the lining up in the middle happens after the kinetochore microtubules are attached, and that's what these are called. They're called kinetochore microtubules because they're locking into that kinetochore. Cells also have non-kinetochore microtubules, which do not attach to the chromosome. Okay, so they're kind of in between and among the chromosomes, and they're overlapping, and we'll talk more about those in anaphase. So metaphase, you've got, this is what it would kind of look like, and the main thing that's happening is you get your chromosomes lined up on that midline. The midline can be called the equator, the midline, the metaphase plate. You'll hear all kinds of terms for that, but it's basically the middle of the cell. All right, then in anaphase, and I'm going to show probably midway through anaphase, or maybe even a little further along. So the cell is already starting to elongate. When the cell starts anaphase, it's still nice and round. But once it gets kind of deep into anaphase, the cell starts getting longer. And I'll talk about how that happens in a minute. All right, my chromosomes have split. So they're these little proteins that hold the sister chromatids together called cohesins. And they have split. The two sister chromatids apart, and so because my kinetochore microtubules were attached on both sides, I'm pulling half of each chromosome each way. If it didn't, then I might end up with too many chromosomes on one side or the other. So we do want to make sure those kinetochore microtubules are locked in on both sides of those chromosomes. All right, then my non-kinetochore microtubules, you'll notice there's very little overlap at this point because one of their main functions is to, let's see if I can show it this way. There we go. They're kind of overlap and they're going to walk away from each other, and as they do, they're pushing that cell, and that's what's elongating that cell is those non-kinetochore microtubules. Okay, so I can describe prophase, what it looks like and what's happening. I can describe metaphase, what it looks like and what's happening. I can describe anaphase, what it looks like and what's happening. Sister chromatids split apart and are moving to opposite ends of the cell. I can describe telophase. I'm going to actually draw telophase and cytokinesis. Whoops. Totally different. Normally I draw them together, but I'm going to draw them different so that you can see what's happening. And so what I'm seeing here, I've got my single chromosomes. My nuclear membrane is starting to reform. Okay, my spindle fibers on my centrioles are kind of breaking down, so they're getting shorter. Basically, everything that happened in prophase is reversing here. So chromosomes are starting to unwind or uncondense. I don't think that's a word, but anyway. Nuclear membrane reforms. You'll start to see nucleolus reform because that also dissolved. Spindle fibers break down. Basically, the cell is remember, mitosis is division of the nucleus. So when we're talking about telophase, we're just talking about now the nuclei are separate, okay? And then cytokinesis, and I'm going to draw it in an animal cell, is, maybe my nucleus is a little more solid. It's getting there. My chromosomes are a little more uncondensed. Again, I don't think that's a word. Centrioles are over here resting because they just did a lot of work. So they're just chilling out. But cytokinesis is not a part of mitosis. It is a part of the mitotic phase, but mitosis is just division of the nucleus. Cytokinesis is division of the cytoplasm and organelles. So when it says I can describe cytokinesis, you're describing that it's the division of the cytoplasm and organelles, and this is what it looks like. We have this pinching in, okay? Now this is an animal cell. Plant cells can't pinch in because they have that really hard cell wall. So the next two kind of piggyback off of this. It says I can compare cell division in plant and animal cells. Ways that it's alike, they all go through PMAT, okay? And that PMAT looks very similar, right? Same things are happening in each one of these. Metaphase, plant and animal, they're both lining up. Anaphase, plant and animal, they're both splitting and heading towards opposite ends of the cell, and that cell is elongating. So there's a lot of similarities. You could say, the kinetochore microtubules attached to the chromosomes, and the non-kinetochore microtubules attached, kind of pair up with another non-kinetochore. All of the little things that we said happen in each of these, you could say as a commonality or a comparison between the two. The contrast or the difference is here. In an animal cell, you have these actin filaments that form, if you can picture like a long balloon, if you tied a string around the middle and you pulled that string, it would pinch the balloon into two pieces or two sections, and that's what's happening here. It's like a filament that kind of goes around, and as it's pulled tighter, it's pinching the cell until it literally pinches it in half. And this is called a cleavage furrow. Cleavage means to separate, and a furrow is like a ditch or a trough, and so it is a depression would be another way to describe it, like a parabola. Is that the right word? Maybe a reverse parabola. Anyway, that is caused by the separation of two things. So that's why it's called a cleavage furrow. Cells, plant cells can't do that because they have a cell wall that's not going to let them squeeze in. And so what they do instead is they have these vesicles full of cell wall stuff, lignin and cellulose and all that, and they line up in the middle, and then they just kind of fuse together and form a new cell wall. And so that's called the cell plate. So that's the big difference in plant and animal cells is during cytokinesis, how their cytoplasm and organelles divide. I can discuss the role of kinetochore microtubules. Okay, those are the ones that attach to the chromosomes, and that is going to serve, they're not actually pulling the chromosomes. Okay, a lot of people think that these are reeled in like on a fishing line, but that's not what happens. What happens is there's this little motor protein. I'm going to show it with a little pink kind of dot, and this little motor protein is going to literally pull the chromosome down the microtubule. So the microtubule, the kinetochore microtubules, these that come in and attach, they are basically like train tracks. They're the tracks that the the chromosome is being walked on. So they're not moving, they're not doing anything, they're just kind of the bridge or the track that's being walked down. So that's the role of the kinetochore microtubules. I can discuss the role of non-kinetochore. We talked about those. They're the ones that are stretching out and elongating that cell. I can discuss the role of kinetochores. Kinetochores are those brackets on the chromosomes where those microtubules attach. And then I can discuss the role of motor proteins. Motor proteins are the little guys that are actually, I say guys, little molecules, that are actually walking the chromosome down these microtubules. If you remember the video where we showed the motor protein pulling the vesicle along a microtubule, it's the same thing, but instead of a vesicle, it's a chromosome. And so this microtubule was out here, and so what's happening is it kind of breaks off into pieces as the chromosome is walked, so the chromosome can't go backwards. It can only go in one direction, and that is towards the poles. And so that's the role of a motor protein. So I hope that helps. That's mitosis really quickly in a nutshell, but it can tell you, it helps you, should help you describe all the phases, what they look like, what's happening in each phase, and some of the differences we see with plant versus animal, and then some of the structures and what are they actually doing in those phases. What is their job? So I hope that helps, but if you still have questions about mitosis, please let me know.